Hello, Mr. Allman here, and this is your Allmancast on the details of transcription. Within the central dogma, we begin with a short sequence of DNA known as a gene. That sequence of DNA, which resides within the nucleus, right now is going to be transcribed into a sequence of single-stranded mRNA, which, when subdivided into sets of 3, can be utilized by the ribosome to be able to produce a unique amino acid chain. You should utilize the stages of initiation, elongation, and termination to describe the basic steps of the process. We're going to begin in initiation at the starting point where it is that mRNA is going to begin. Now, the DNA is extremely long, and only a very small percentage of the DNA is actually going to get transcribed into mRNA. So, the enzymes responsible need to have some way to know where to begin and where to end. Now, the process of initiation is going to begin when RNA polymerase, the enzyme that is going to make the RNA polymer, when it attaches to a region of DNA known as the promoter. That promoter is going to be slightly in front of the very beginning of that gene to ensure that nothing is skipped in the beginning. Now, in eukaryotic cells, transcription factor proteins are going to be bound to that promoter region of the DNA. So this green sequence is going to illustrate here the promoter region, and there is a certain sequence of DNA to which transcription factor proteins are going to be attracted to and to bind. Once those transcription factor proteins are bound to that set of DNA, then that's going to recruit other proteins, including the RNA polymerase molecule, which are then going to form what's called the transcription initiation complex. In other words, there's going to be a big collection of many proteins that are going to work together to be able to transcribe this particular gene. You should be aware that one specific function of RNA polymerase is to separate the two strands of DNA from each other, breaking the hydrogen bonds between complementary base pairs in order to expose the nucleotide bases that need to be transcribed into mRNA. We continue then with elongation. In elongation, we're going to produce a strand of mRNA that is complementary to the DNA template strand. You can see here how the blue strands of DNA, the non-template and the template strand, have been separated, and we have the enzyme that surrounds that bubble. Now, transcription is happening in this particular image from left to right, but that terminology is not really helpful for describing the orientation within the cell. So instead we need to refer to the ends of the DNA based on their sugar phosphate backbone. We have a template strand of DNA, which is 3' to 5'. So the darker blue is the template strand of DNA, and that is going from the 3' to 5' direction, which means that as mRNA is built in the anti-parallel to that template strand, mRNA is being built from 5' to 3'. RNA polymerase simply moves in that direction towards from 5' to 3' as it builds the mRNA, adding on RNA nucleotides that are free-floating in the nucleus in the area surrounding the enzyme, adding them based on the complementary base pairing rules for RNA. This process continues for the entire length of the gene. Once the end of the gene is reached, then termination is going to be when the mRNA strand detaches from the DNA template. RNA polymerase detaches, many of those transcription factors uncouple from each other, and now that mRNA is free to be able to be processed and leave the nucleus. Now, lots of things actually do get transcribed. though this video is focusing on the function of transcription in terms of making mRNA to build proteins, actually transcription can happen to produce structural forms of RNA, such as the structural RNA that's used to make tRNAs for translation, and the structural RNA that's used to build ribosomes, which are also going to be useful for protein synthesis. Now we're going to look at what happens to that mRNA, because it's not yet ready to leave the nucleus, as we look at the process as it occurs in eukaryotic cells. So post-transcriptional mRNA processing, which does not happen in bacteria, but does happen in eukaryotic cells. We have three sets of events that need to happen. The first is that things are added. We have a 5' GTP cap. The GTP cap, G standing for guanine triphosphate. So this is a modified guanine nucleotide, and this is added to the 5' end. This is going to be important in directing that mRNA sequence through the nuclear pore and out to the ribosome, where it's going to be read. At the end is going to be the poly A tail. There is a long sequence of adenine nucleotides added at the end of the mRNA chain. These are never going to be translated and never going to result in amino acids being added, but they do serve an important protective function, because throughout the nucleus and cytoplasm there are many enzymes that are responsible for chopping and breaking up a used strand of RNA. So the presence of that poly A tail is important to ensure that if those enzymes come and chop off some little nucleotides at the end of the strand, they're not going to affect the important protein coding segment, which is found within the middle of that chain. Splicing is going to be the second important thing that's going to happen. A sequence of DNA is going to have these kind of alternating regions called introns and exons. The region that is going to be transcribed and should leave the nucleus and be translated is called the exon. Those coding regions of DNA, those exons, they need to exit the nucleus. So those need to stay in the mRNA in order for them to then be ready to encode for an amino acid. The introns, or the part in between, they need to stay in the nucleus. So those introns are sections that need to get spliced out. So there is a spliceosome, an enzymatic RNA molecule that is capable of recognizing particular sequences of DNA that show the beginning and end of a particular intron. They will cut the RNA strand at the beginning of that intron, they will cut it again at the end of that intron, and then join together the two ends, discarding that extra intron, that extra strand of RNA, which is no longer needed. So those introns all along the length of the mRNA, whether there be one or multiple of them, they need to get cut out, the ends spliced together so that what you end up with is the cap, the coding region of DNA consisting only of exons, and the poly A tail. Now, those introns get removed, but they don't get removed every single time. One of the functions of those introns is in order to allow for alternative splicing. If a given region of DNA has multiple different exons, at times those exons could get recombined in different or unique ways. If we have exon 1, 2, 3, 4, and 5 is just off of the picture, we can splice those together in different ways in order to be able to show proteins that vary slightly. If we look at our two central pictures here, if we have 2, 3, 4, and 5 put together in this order, then we get the blue, purple, the peach, and the pink regions. Then, if we have a second version of that protein where exon 3 was spliced out and not left in, now we go from the green region, which was not present there, to blue to peach, skipping the purple, which was exon 3, and so now we've made two different proteins from the same piece of DNA. The more exons you have, then the more different ways in which that those exons could be spliced together, giving you a chance to be able to make a unique protein from the same set of DNA. This helps to explain the reason why it is that even though we have maybe close to 30,000 genes, we make nearly 100,000 different proteins. What that means is that it's not one gene per protein, but we could have one gene per 2 or 3 or 5 different proteins, thus increasing the variety of proteins that a living thing could make without necessarily having to increase the amount of DNA that it carries. That's all for the details of transcription. Look for the next video on the details of translation. Thank you, and let me know if you have any questions.